Thank you for joining the Once Changing the World, India's first Future Tech Meets Sustainability podcast. And today, I'm delighted and honored to have with me Dr. Hongkui Deng, who's a leading immunologist and a global leader in stem cell research. He received his PhD in immunology from UCLA before switching gears to focus on stem cells at YSL in Boston. Dr. Deng's groundbreaking research earned him a $1.9 million grant from the Bill and Melinda Gates Foundation to develop vaccines for HIV and hepatitis C. Dr. Deng and his team used CRISPR gene editing to create HIV resistance in mice. This research culminated in the first use of CRISPR on a human HIV patient. While the treatment wasn't a complete success, it demonstrated the safety of CRISPR technology in humans, paving the way for future advancement. He later relocated to his home country, China, and is currently the director of Peking's University Institute of Stem Cell Research and has received the Boya Chair Professorship. Dr. Deng's other revolutionary contributions is the creation of human chemically induced pluripotent stem cells. And Dr. Deng was recently also awarded the Future Science Prize 2024 in Life Science. He is currently in the news for reversing Type 1 Diabetes via the patient's own reprogrammed CIPS stem cells. So, Doctor, really appreciate you taking time and being part of the podcast. It'll be great if you can, talk about your human HIV patient, experiment or research with CRISPR. Where does the research stand currently as of now?
Okay, yeah. So I have been working in that field for many years. Actually, it started 30 years ago, like 30 years ago. So when I was doing my postdoc research at NYU, New York Medical School University. At that time, my supervisor is Dan Littman. He's a famous immunologist. So when I was doing my postdoc, at that time, scientific question is how to try to find the receptor, the viral receptor on the cell surface. So the HIV virus can bind to receptor and get into the cells. At that time, we tried a big effort to try to identify the critical receptor for HIV. And we published the paper in 1996. That's like 28 years ago. So we identified the major receptor is called the CCR5. So that's the key receptor for HIV getting into the host cells. So after that discovery and many interesting, important research following that research, for example, in human population, particularly in Europe, in some people that don't have this receptor by nature. So these people are resistant for HIV infection. So this famous Germany doctor, he took the cells, the donor, from the person that don't have the receptor for this. So this one famous HIV patient, he has HIV and leukemia. So traditionally for leukemia treatment, you need a bone marrow transplantation. So this famous Germany doctor, he chose the donor, and the donor has don't have the CCR5, the gene expression. So he smartly transplant this cell into the HIV patient, and this is this HIV patient we call the called the burning patient, famous. So he was the first. He's the first person was cured for HIV treatment, just simply by hematopoietic stem cell bone marrow transplantation. And the donor has a CCR5 mutation. So this is a famous case. Actually, following that publication, now they have 6 patients treatment like that, all being cured. So it demonstrated 2 things. So this bone marrow transplantation approach from this kind of donor is successful. It's not only unique for that burning patient. So it's right now, so far, I have 6 patients' data to report it. Secondly, emphasize the CCR5 is ideal the drug target for cure for HIV. So that's the that's
Right.
the background for this work. So in 2017, so that's 7 years ago. So we started the first clinical trial. So we used the bone marrow transplantation, but this cell is a normal cell. But we use CRISPR technology to knock down CCR5. And then after the treatment, we put back the bone marrow cell into the patient. So the patient is the first CRISPR treatment for this kind. Because but this work is not use like an embryonic cell. This is adult hematopoietic stem cell. So we don't have ethical problem, right? It's not doing embryo research. It's doing like adult hematopoietic stem cell. I want to emphasize for that. So this is we don't have an ethical problem. So for this.
Right.
So this patient has been treatment now is 7 years, right? So we published the work in 2019. So because we started clinical trial 2017, 2 years later, we published that result in the top medical journal called the New England Journal of Medicine, reported that case. It tell you the feasibility, safety, for that approach. So we should say it just says promising. We need a continual effort for that. So we are continuing for that because at that time the CRISPR technology cannot knock down CCR5 100%. So only knock down the 30% in some extent. So we continually improving that technology. So in someday, if we can completely knock down CCR5, that will be like the breakthrough approach in the future.
Right. How cool is
Sorry,
that?
I take a long time.
No, not at all. I mean, doctor, thank you for explaining. And I hope that, this technology kind of evolves to a point where we can actually eventually knock down the CCR5 gene, and then how cool that would be, So, I also want to get into, the iPSC versus CPSC. What are the key differences between the Yamanaka's approach to creating iPSC and chemical reprogramming?
So in the future, traditionally for biotechnology, we use small molecular like chemicals. Like if you go to see doctor, they either do surgery or give you some kind of chemicals for like drug, like medicine. And now last 20 years, we have a big called antibody, right, treatment. But in the future, this very promising field is called cell therapy, or you call it regenerative medicine. So if in the future we can use the regenerative medicine approach, you can do the cell replacement therapy for people because of age, disease, injury, or genetic defect. You can replace the diseased cell or injured cell with a normal cell, right? That's the future. So for this future medicine, the behind this, the fundamental technology is called reprogramming, called the cellular reprogramming. What do we call the cellular reprogramming? Because all the human body, the cell, the skin cell, hair cell, blood cell, all come from because the first cell fertilize the egg, right? And then you start differentiation, generating many different specialized cells. Because this all different specialized cell, you have the same genome, but it's just because their functional specialized become different type of cell. In principle, they can reverse back, developmentally reverse back to early stage. This reverse process is called reprogramming. Okay? So in the field, in the last 40 years, these are top question in the stem cell field. How to reverse them back? How to reprogram the cell become an early embryonic cell stage? So the first generation technology used the egg cell. Like people use the egg cell, the clone the dolly, the famous clone the. So if you take some kind of somatic cell, for example, monkey cell or sheep cell, you take the nucleus from the cell and you do the microinjection, put them into enucleated egg. And the egg cell has the trick, can reprogram itself. And you can reprogram it back. Erase all the specialized, the developmental, the modifications. They can reverse back to the early point, and then you're generating cloning. That's the cloning technology. So in 2006, this famous Japanese scientist called Shinya Yamanaka, he did a groundbreaking research. He found you don't need an egg anymore. He just put 4 transcription factor, the key gene controls the cell fate. You just simply transfect or transduce this gene into the somatic cell. You can reprogram cell, bypass the egg. So this is called the genetic approach. You just enforce the cell expression, this 4 key, we call the transcriptional factors. You can reprogram yourself. So he got the Nobel Prize 2012, right? Famous word. But we call this second generation technology. The first is use egg. The second one is use a genetic approach, right? So what's the third one? We're developing the third generation. So what is the limitation for first one and the second one? So first one, second one is either you depend on egg or you depend on these genetic factors, right? So this reprogramming process, you cannot control this process. You enforce the cell expression of these genes, and then all the way back become the pluripotent stem cell. If you take an elevator, this non-stop from the first floor directly go to 15. You cannot stop like a 2, 3rd. It's not a controllable process. So we want it to become like you can control them, precisely control. You can stop at any kind of stage, and you reverse them back. So that's what we are interested in. So first. Secondly, in some lower animal model species that have remarkable regeneration capacity. I'm not sure you know the axolotl.
Yes.
There's many. You can cut any part that can regenerate very robustly. But human body, we cannot, robustly. If you lose the arm, you cannot regenerate. But in the lower animal model species, they can regenerate. Right? Then you can regenerate very robust. The question is why? Why the evolution we lost this capacity? What's the trick? Right? So we want to learn from this lower animal model species because we think this is a better pathway, can mimic the lower animal model regeneration process, and we can make this process become much controllable. Right? So that's what we call the chemical reprogramming. So chemical reprogramming is very simple. You just put the cell in the culture solution, the solution containing chemical. You don't need a special equipment. You don't need a special technology. You just put your cell into this solution. The solution has a chemical cocktail, and they can reprogram themselves. That's this technology. And this cocktail, you can change their concentration, treatment time, and you can make a different cocktail, right? So you can change the cell fate under your control. Wow. So that's the principle behind this. So we work on this for many, many years. So we published the paper 2013, that's 11 years ago. We demonstrated in the mouse. If you use the mouse fibroblast, skin cell, you can reprogram it all the way back, become a pluripotent stem cell. But it's so hard for human cell because human cells has much more, we call the epigenetic barrier. So it's more difficult. So we spent 9 years. So finally, 2 years ago, we published the paper. We can do human cell now, very efficient. So we call reprogramming technology is the basic and the fundamental technology. You can change the cell fate change the cell identity, change the cell functionality. So it's like a CRISPR, you can edit in human genome, but chemical reprogramming, you can like a reprogramming, you can also call the editing the cell fate, cell identity. So this is a powerful technology.
Wow, wow. Doctor, how cool is this? Because I think, we're getting closer and closer to possibly, I don't know whether it's the right word, but maybe to becoming the creator ourselves. Where yeah, where because I think yes, I'm sure it's still in a very nascent stage, but if this technology, whatever time it takes, for it to to evolve, I mean, to actually be able to, like you said, to change the cell's fate or identity or functionality, what could that mean for the future of humanity? It could mean from
So,
age reversal to diseases going, there are more
so,
diseases. That
so,
would be, yeah,
yeah. So yeah, continue. You you
yeah,
finish it.
yeah. No, yes, I think that would be a beautiful, beautiful world. Because a world is so divided when it needs to be united. Where
Yeah.
I think the pharma companies, maybe because of the billions of dollars they kind of invest into creating a drug, and the VCs who have obviously, because there's so much of money that they invest, their their only, I think, goal is to maximize profit rather than to create drugs which are accessible for everyone. I, I mean, it's what a world it would be if we are able to control the cell's fate, identity, and functionality. Uh, Would there be a disease? If we get into a world like that?
So we are working on technology for future medicine. That I'm not talking about the now. We are, it's promising for the future. We need, to really, we have the science is the drive for new medicine. So that's for the future. So the three possibilities. The one possibility in the future is we can generating functional cells in vitro. So we can generating like a recent example, we're generating insulin producing cells, right? And then you put because some people insulin secreting cell in the body are dysfunctional. If you can generating insulin producing cell, you put it back to patients to help them, right? So that's the one dream we try to do. We can do cell therapy. We can generating functional cells in vitro. Maybe in the future, maybe for tissue or organ, we can generate in some way, can do the, organ transplantation, but that's far in the future, need a huge amount of work. Secondly, in the future, maybe in some disease, we can regenerate in vivo, right? Just like a lower animal model species, you can regenerate yourself. It's not in vitro. You make some cell and transplant. Maybe just in vivo you can regenerate some cells, right? And thirdly, in some disease, we can maybe reverse the disease, the phenotype, right? You can in some disease early stage, maybe you even can manipulating and reverse them, right? Some way. So but that we all talk about the future.
Ah, right, right. No, no, no. So because the future is so exciting, but
Yeah,
yes,
yeah,
let's come to the present. Like the
yeah,
present is, can you explain your recent
yeah, yeah,
breakthrough in
yeah,
using stem cells
yeah,
to treat type 1
yeah.
diabetes?
Yeah. So recently the case, we actually work on this for 23 years. We started work on this in 2001. So it's a long time ago, right? At that time, we don't know it take how many years. So basically, it's the three technology combined together. So first, we need developing reprogramming technology. So we call it chemical reprogramming. I just explained to you. So we actually start 2001. We also work on if you have reprogramming technology, if you have pluripotent stem cells, in principle, you can use this as a seed cells, right? Starting material. and make a direct differentiation, generating particularly differentiated specialized functional cell. So we try to generate insulin producing cell. So we actually published first paper 2005, almost 20 years ago. But that time it's for mouse. We always start from the mouse cells. So 2007, we published first paper human cells. So we try to generating human insulin producing cell. And people say, oh, we call the insulin producing like cell because a little bit like insulin produce cell. Because the only can produce a very small amount of insulin. The level is 1004, lower than normal insulin producing cell. So that's the early stage. So we produce the insulin producing like cell and getting much like cell, and then get to become real like cell. Take how many? Take 20 years. It takes a long time. So finally, 2 years ago, so we really made the really insulin-like cell. So we first test in the monkey model. So the monkey has the monkey diabetes. If you put the cell back, and you don't need the insulin injection, you treat. Yeah. So so we did in the mouse, and then did the big animal model monkey. We tested this, and then we need to find a way to put the cell back. So that's the third technology, because the insulin producing cell is very special. You don't need to put it back in situ. It's not like a neural cell. You need to put it back exactly where the destroyed site, right? Heart, you need to put it back to heart. You cannot put it like into other part. But insulin producing cell, because they are just sensor glucose and then produce insulin, in principle, you can put any kind of part in your body. You just two requirements. They can sensor glucose change, and then they can produce insulin and release into the blood, and then you can go to the target organ. So that's a special case. We need to find where to put back the cell, the best way. And then we find this site. It's a little bit difficult to say the name. We published the paper. It's called anterior rectus sheath. This place, you need to have space. And you put the cell can survive there. And the cell, you cannot move to other place, encapsulated there. So first, that has space can take the cells. Secondly, the cell being encapsulated there. So now like move to other part, right? So you have to stay there. So thirdly, the cell can be survival there, long-term survival there. The vascularized the survival there. And the most importantly, we can monitoring cells there and see how safe. You can usually by ultrasound, MRI, you can see the cells, see if they are normal or easily can check. If you put some powder, you cannot find them. So this is for safety reason you put them. And finally, the surgery is very simple. It's very easy. It's a small surgery to put it there. So because of this several requirement, we found this ideal site to put it there. So we combine the three technologies. So reprogramming, direct differentiation, and the transplantation strategy all together. So we demonstrate the monkey first. We can cure monkey diabetes. So finally, we get the government approved. So you can start a clinical trial last year. So we tried the patient. So after put back in, just like in the animal model data, after 75 days, the patient become insulin independence and maintain the insulin independence for 1 year now. So patients, everything is back to, so we reverse back to normal level.
Right. That's
just for prove the principle. This is okay.
So congratulations, doctor, to you and the entire team for doing this in the most rigorous way as possible, right from
Yeah,
testing it out on mice, then,
yeah,
monkeys, and then getting on to human trials.
yeah.
So what's the next step? So this is a lady that, this experiment
Yeah,
was done on. What is there further clinical trials that's going to
yeah, yeah,
happen?
yeah.
And what would be the next step in bringing this treatment from the
So
laboratory to actual clinical
the three
practice?
things. First, we need to try more patient, right? Well, you need to demonstrate more patient, and we have to follow up the patient for a longer time. So now the patient is 1 year and 3 months. So we want to say 5 years later, 10 years later, you want to become long-term safety, and so you need to take time. And then we need to try more patient. We need to try type 1, and then we need to try type 2. And more importantly, we need to make the cost less, right? We need to make this much reduce the cost, and then you can access to more people. So there's many things to do.
Right, right.
Yeah.
So
Just to prove the principle, this is you can do. I don't know the cell phone. Like the first cell phone, I don't have cell phone. At that time, maybe very expensive. I mean, right at the beginning, right? You can optimize the technology, and to much. We need to take a big effort to optimize this. Become a second generation, third generation, treat more people. That's the future. And then we need to importantly follow the patient for long term, right? To say
right.
the
Right,
yeah,
right. So congratulations, doctor, because I think it's such a groundbreaking proof of
yeah,
concept that this has been done. It's proven with the patient, and
yeah,
you are going to now continue with more clinical trials with more
yeah,
patient.
yeah.
Can you still maybe possibly elaborate on the future direction? What's your insight on this? When do you think, because you said obviously, there are people who say that you need at least 5 years to kind of, test, uh, keep on understanding the progress of the patient. What's your assumption on this? Because there are millions in the world, possibly who suffering from the diabetes. Type 1 and
Yeah,
type 2. When do you think this could come from the laboratory to actually start helping patients worldwide?
yeah, good question. So I think we need a international collaboration. The collaboration is the key. So we can make the technology widely, to be applicable to by accessible for more patients. So firstly, I think at this stage we just prove the principle of like reprogramming technology, chemical reprogramming, right? We need a better chemical reprogramming in the future, and then we produce insulin like the cell. In the future, we'll make them maybe identical, not alike, it's identical to real insulin producer. We still need it. In the future, for this transplantation technology, we can still optimize the process, right? In the future, we don't worry about the safety. If any safety, very simple surgery, just take out. So we put the bar for safety bar much higher, so people never worry about it. So this is a safe technology, right? We need that, and then we need to reduce the cost, and we need to try more patients. It's just and
Right.
do internationally, to many hospitals, and worldwide, everybody. So we tried this. So this is this is the end, not the end. It's just beginning.
Beginning,
It's
right?
really at the beginning.
Right. Congratulations on the
Yeah,
hard work that you've put
yeah,
to kind of, bring it this far. And I hope that this, goes out and touches the lives of those various billions of people around the world who are suffering, because of this disease. Doctor, my last questions to you.
yeah,
Uh, how do you see this evolving in the future? Because obviously you gave us an insight on, these chemically
yeah,
induced pluripotent stem cells.
yeah,
It could help in regenerative medicine. It could help in aging
yeah,
research. And I also read that, it could help in disease modeling, drug discovery.
yeah, yeah.
How do you see this impacting the future of healthcare? So it would be great if you could paint us a picture of what you think this could do in the next, the future of healthcare. What are the
Yeah, yeah.
challenges, and
I,
what are your
I I think
plans to overcome that?
I think the thing is the most fundamental is chemical reprogramming. So I'm sure this will become technology for not only for diabetes, for many other diseases. So you can use this reprogramming technology. We still need to improve this, but in the future will become you can become like, I think use the reprogramming technology to manipulating cell fate, cell identity, cell functionality is becoming general, right? And then after manipulation, you can use for not only for cell therapy, you can do study, like do research, understand the disease process. You can use this disease model, use the in vitro as a disease model. You can do drug screening based on this. So not only just for cell therapy. So so we can widely use this reprogramming technology for the future.
Right. Doctor, really appreciate you taking time and being part of the podcast. I think we're sitting in a cusp of a future which could actually democratize and make healthcare accessible for everyone, and where we could possibly reverse age. We could.
I hope.
We could possibly maybe cure so many diseases. So thank you for doing this. And to my listeners, if you like what you see in here, then please press the subscribe button. Until next time, see you guys. Bye-bye. Thank you, Doctor. Really appreciate it.
Thank you. I really enjoyed talking to you.
Thank you, Doctor.